Okay, so hello everyone. I'm very happy to be here. I am Charlotte Moritz. I am a consultant in research based in French Polynesia, and I work with the different research institutes there. First, I would like to thank Luminocean and especially Mareike, Joanna, and Priska for organizing this workshop and inviting me to present my research. So my presentation is about coral spawning as well, and it is entitled Knowing Before Restoring, Shedding Light on Daytime Spawning in Porites Rus. Yesterday, Jeremy Craggs talked a lot about coral spawning, especially corals that spawn at night. But here today, I'm going to present you one coral that spawns during the day because this happens too. So you have a picture at the back of the slide showing a branching Porites Rus, and you see a white cloud coming out of the coral, and this is the sperm. So this is a male Porites Rus that is spawning. So as you all know, currently there's a general trend towards global decline of coral reefs, which is very concerning. And this is why we see restoration as a potential small-scale solution to restore reef at specific sites, especially nowadays because there are many new material, new techniques, and there are more and more people involved around the world and trying to do restoration in many different places. Restoration has made big improvements over the last decade, as we saw yesterday and today with Jamie, Tris, and Vincent. Now we can even produce larvae from adults, and we can also produce baby corals from the larvae produced from the adults and make them settle and restore the reefs. What has been presented today is mainly about very well-known corals such as Acroporas, Pocilloporas, etc. But what about understudied corals? I'm not a big expert in restoration, so I'm not sure how many species of corals have been gardened, but for sure not all the species have been gardened yet. So there are many more things that we can learn about other coral species for restoration. So it's very important to continue monitoring, to continue gather knowledge of all coral species to better cultivate them and restore them. Especially we need to know the effects of the environment on them because it's very important for their growth and their maintenance in a specific site. And we have to know about their reproduction because as you saw yesterday, the corals can have different reproduction types. And they reproduce at different times of the year, of the lunar cycle, and at different times of the night or the day. So usually the coral spawning is synchronous and massive among species or within one species. It is very well known for nighttime species. So as Jamie talked about yesterday, there are a lot of works going on with the Acropora species, especially to better understand the spawning timing, the synchrony, the triggers that induce gamete release. But it's much less known for daytime spawning species, although there are many coral that spawn during the day. So what happens is that you have environmental signals that induce gamete maturation in the polyps. These gametes are released in the water on the spawning day. So you have male and female gametes that mix in the water. You have fertilization going on that forms a larvae that can disperse afterwards with the currents and make recruits for sites more or less near the spawning.
Rekrut di beberapa site.
There are actually 3 timescales over the year
Dan ada 3 skala.
So the first scale is at the whole year long. So you have the annual temperature cycle, when you have the seawater and the air temperature that warms up during spring and summer towards the warm season in the tropics. And you have the photoperiod that changes over the year with longer days, and that will determine the beginning of the spawning seasons in corals. This induces gamete maturation over several weeks to several months. And then you have a second time scale, which is smaller, which is at the time scale of the lunar cycle. The nocturnal light is detected by the polyp, and this will determine the spawning date depending on the species. So the coral prepares itself to expel the mature gametes. And finally, you have a third time scale, which is even smaller. It's at the day or the hour scale, and it's about the amount of light or dark after or before sunrise or after or before sunset, and according to moon or sun, depending if you have a nighttime or daytime spawning coral. There are many different triggers as well, actually, because every species can react differently to these environmental cues. Yesterday, we saw with Jamie that, for instance, the importance of the period of dark after sunset was very important and could change according to the species. So this determines the exact spawning time when the gamete will be released from the polyps. So I'm going to present you the main results that are described in this paper that finally came out last June in Global Ecology and Biogeography. It's the work with a collaboration with a non-profit organization based here in French Polynesia that I will present in the next slides. So some of you have already seen these results during the workshop in January with Luminocean. And for the other one, welcome, and feel free to ask any question at the end of the presentation. So how this started, why did we start studying Porites Rus in French Polynesia? It's because the first observation was a strange white cloud, as you can see on the picture on top of the statement, near Porites Rus in November 2014 in French Polynesia. So we didn't know what it was. And then a same cloud was observed from the surface, actually, in November, the same month, November, but in 2017. And another one in November 2018, underwater. So we made a little bit of search on the internet, and we found this publication from Bornstein and Loya, who recorded daytime spawning of Porites Rus the fifth day after full moon. So DAFM here is for day after full moon. And this is actually exactly the same as the observation that we had in 2014, 17, and 18. So we tried to make a prediction for 2019. So 5 days after the full moon, about 1.5 hours after sunrise. So it was the 7th. We predicted that Porites would spawn on the 17th of November, and it actually worked very well. The prediction was very accurate, and we saw the Porites was spawning. And we did the same for 2020, and it worked as well. And actually, we were wondering if there was really only 1 month of spawning for Porites Rus over the year. So we went in the field and did the same observation in December, 5 days after the full moon, and the same in January, February. And actually, we saw spawning every month until April. So this allowed us to determine that for Porites Rus in French Polynesia, the spawning season is quite large. It extends from October to April. So it's over the warm season in French Polynesia, which is also the rainy season. What we can see on this graph here is that the spawning time is really related to the sunrise. So the coral will spawn almost exactly 1.5 hours after sunrise. However, it's not correlated to moonrise the night before. Maybe there's some cue with moonrise, but we haven't detected them yet. So after this observation, some of the observers decided to create an organization to share this knowledge and this observation with the public and with the pupils here and the students here in French Polynesia. So they created the association, the organization called Tamano Tetari Roto, which means the children of the lagoon in French Polynesian language. It was created in the end of 2021, based in French Polynesia. You have the address and the Facebook page on the right of the slide here. And at the same time, they created an observation network that everybody can join called Yaura Tetoa, which means long live the coral. And they started to organize observation sessions for the population. So everybody could go with us and observe the spawning of Porites Rus in the field. And we gathered quickly a lot of observers that were able to collect and provide us the data of the exact spawning time and day for Porites Rus around French Polynesia. The organization also communicates on the marine environment. They make talks in schools, etc. They implement projects with colors. And they have created a web app to record the observation. So you can go on the web app that is called Tamano Tetairoto. You subscribe to this app and you can record your own observation. So even it works around the world. On the app, there's a world map. So you can just locate your observation, even if it's in Indonesia, and you can record your observation. You can record your observation for Porites Rus, which is our main target, but you can also record observation for other species. And this is through this work with the organization that I came across Marika and Joanna from Luminocean, and we start talking about coral spawning, and they invited me to join their network and workshops. So you have another picture here of a male Porites Rus. What we have to know is that it is a gonochoric species, so you have male colonies and you have female colonies. So this is a picture of a male that is spawning, and you really can't miss it because the cloud is very, very dense and very white and bright in the water. What is very interesting with Porites Rus, it's a very, very resistant coral, which is very good to study in this context of global change and climate change. It is highly resistant to temperature. High temperature do not affect it. It is very resistant to UV light. It can grow very close to the water and is not really affected by UV. It is also resistant to sedimentation. You can have it close to river mouth, close to the beach, and the sedimentation does not affect it much. You can have it in several current conditions. You have the encrusting morph that can grow in very strong current conditions. So this is why researchers in the coral reef research area often call it a winter coral. It easily adapts to different stressful conditions. In French Polynesia, it is very close to the coastline, which and it's highly abundant. I'm not sure in Indonesia, but it depends on the countries. Even in the Pacific Islands, in French Polynesia, it's very highly abundant. In New Caledonia, for instance, it's much less abundant, but there is also some Porites Rus there. So it's almost everywhere. And the fact that it is close to the coastline, it's really close to the beach. You can just walk there and you find Porites Rus very easily. It is good for the observation that we do with the organization using citizen science. What is very interesting is that it can grow from 0 meter from the coastline, very shallow waters, to more than 80 meter deep. So it can grow at a very large range of depths. It also has different morphologies, as you can see on the pictures there. You have a picture where you have a branching morphologies, massive morphologies, encrusting, and plating. Usually, when you increase the depth, the Porites becomes more like a rose, like rose petals with a plate morphology. We think it's to better capture the light, because when you go at greater depths, you have less light, and the polyps need to get this light to grow. So with the organization, we started to collect data using citizen science and with the help of the observers from the population, from the marine institutes, from the organization. Everybody who wanted to join could join. And we started to build 2 data sets. The first data set is a site data set, which means that we are going to record the first colony who spawns within this site. So we record the exact time at the exact minute when the first colony we see is spawning. The sites are usually about 50 to 50 meter large and long. It needs to be not too big to be able to be monitored by 2 or 3 people. In the publication that I'm going to show you right now, we had 15 islands recorded. We even have a bit more now, because we have gathered more and more people around the world. We have colonies that are located from 0 to 30 meter deep. So at 0 meter, you can just go snorkeling, observe the spawning, whereas below 5 to 10 meter deep, you have to go scuba diving. So this site data set has started with the first observation I mentioned before in 2014, and it's ongoing. And with the organization, we decided to collect data at the colony level. So we tagged a few colonies in several specific sites. So it's a more scientific approach, which is much more accurate. And we only take very experienced people to collect the data from these colonies. At present, we had 125 colonies. We have a little bit less now because some fell off, some other bleached, and some sites we didn't have enough people to help monitor all these colonies. So we have a little bit less nowadays. These colonies are also located between 0 and 30 meter deep. And we started this very accurate monitoring in 2021, and it's ongoing. And for this specific monitoring data set, we also record if the colony is a male or a female. So here you have 2 pictures, one that I showed you on the slide just before, which represents a male colony that is spawning. And the second one is a zoom of a female colony that is spawning. It is a bit more difficult to see because the eggs are very, very small. They are rounded. They are slightly pinkish. and they are very small. They are less than 1 millimeter diameter. So you have to go very close to the coral to be able to see the female spawning. But when it is intense, you can really see these eggs, pinkish eggs going out of the corals and floating to the surface. What we noticed when recording this, the time for each male and colony female is that the female spawn on average about 20 minutes later than males. It depends on the sites, but usually it's between 15 and 20 minutes after male. So in one particular site, you will usually always have the males that start spawning, and then the female will start about 20 minutes later. So what's new in the world of coral spawning research with our monitoring of Porites Rus? This is a map that shows the exact decimal time after sunrise, and you have every yellow circle is one island, so you have many islands that we monitored in French Polynesia. You have some observations in Fiji and in La Réunion, and since then, because the yellow bubbles are from the papers I'm going to present you today, but since then we have added a little bit more islands from Indonesia. You can see the star on Indonesia here, thanks to Marijke and Joanna and Luminocean's work, and at some other places where we found the same spawning timing and synchrony for Porites Rus. So you can see in the bubble, the times are really similar between the islands. It's always after around 8 o'clock in the morning after sunrise. So here you have a more detailed map of our site. So we had one or two observations in Fiji, one in La Réunion, and several ones in French Polynesia, especially in Tahiti, Moorea, and the Society Islands. What we observed is a really good and strong consistency in spawning time at all islands that we monitored. Here on the graph below, you have the decimal spawning start time after sunrise, and you can see that at all islands, the spawning start time is really close to the blue line, which is the average across all islands. So there is only little variation around this average. You have the exact data and all data indicated on this graph. It means that the exact data is when people can record exactly at the very minute accuracy the time of spawning for the Porites. And we also record the approximate data to be able to gather as much information as possible. But sometimes the people are not really sure when the spawning started, or they missed the spawning very start, so they cannot record the exact data, and they record the approximate data. But this graph shows that we can also use the approximate data. They are still quite good for the analysis. The spawning time is also really accurate at the site level and at the colony level. Here you have the Veirao site, which is located in Tahiti, and it's one of the sites that is monitored with specific tagged colonies. And what we can see here is that the spawning time, you have two groups of blue diamonds on the graph. It corresponds to two different spawning seasons, 2021-2022, from October to April, and from season 2022-2023, from October to April. And we see only little variability within one site in the spawning time of all the colonies. Here you have specific colonies. So each dot, each color represents one colony, and you see that the average is the line, the colored line for each colony. And still at the colony level, you only have very limited variability around the spawning time, which means that every colony is like very, very accurate every year to release its gametes. Of course, there is some variability around the mean, which can be due to the inter-individual variability. Of course, it's a biological organism, so you always have inter-individual variability. And maybe this can depend also on geographic and environmental site characteristics, because you have a variety of islands, a variety of habitats, a variety of environmental conditions in the different sites, and this can also induce some variability in the spawning time. So the influence of depth, as I mentioned just before, when presenting the data sets that we have, we made the observation at in shallow waters between 0 and 5 meter deep, and we made observation below 5 meter deep and down to about 30 meter deep. Here on this graph, you have the decimal time of sunrise and the exact decimal spawning start time. We can see that for all these observations from the shallow lagoon, there is a really good correlation between the time of sunrise and the spawning time. So the Porites is really, really regular when spawning after sunrise. When we add the data that we observed for the four reef, the outer slope and the channel area, which are around 20 meter deep, so the data in red on this graph, we observed that the spawning was actually much, much later, about 3 hours after the spawning in the shallow lagoon, around 10 a.m. Well, in the morning. So there is a big difference in spawning start time when you change the depth where the colonies are located. So we wanted to see if there was a correlation between colony depth on this graph and the decimal spawning start time after sunrise. And what we observed is that, yes, there is a linear statistical significant correlation between depth and spawning start time after sunrise, especially when, well, we have at the moment a lot of observation at the shallow depths between 0 and 5 meters, and a lot of observation around 25 to 30 meter deep. But we have much less data, and at the moment, mostly approximate data at intermediate depths around 10, 15, 20 meter depths. So we do have a linear relationship between depth and spawning time, which means that at greater depths, the colony of Porites Rus will spawn much later. But there is still some variability at intermediate depths around 10, 20 meter depth. So I think we need to gather additional data to better understand what happens here in this correlation. Maybe we don't have exactly a linear model. Maybe there's a strong variability at intermediate depths, maybe not. So I think there is more work that needs to be done with correlating depth and spawning time at these intermediate depths. So because we saw just before that when you go at stronger depth, you have less light. So we think that if Porites spawns later at greater depth, maybe it's because there is less light that can trigger gamete release. So we wanted to investigate that, and our hypothesis was that the polyps need to accumulate enough light after sunrise to induce gamete release. So we installed in two sites at 2 meter deep and at 20 meter deep, one PAR logger, which means photosynthetic active radiation logger. It's a logger that records the light every day, every 10 minutes. And we placed these loggers at the beginning of the spawning season, 1 year, and we removed them at the end of the spawning season, and we had the light data for the whole spawning season. And what we did is that we calculated the light that was accumulated between dark, so just before the sunrise, and between the spawning start time. And we did that for each colony that was surveyed and at each date that was available throughout this spawning season. And we obtained this graph here. So on the x-axis, you have the cumulated light that is captured by the spawning and by the polyp between dark and spawning start time. And on the y-axis, you have the exact decimal spawning start time after sunrise. What we observed is a correlation between accumulated light and spawning start time. So when the light, the accumulation light is high, the colonies tend to spawn earlier. There's one exception at the first spawning of the season. What is very strange with Porites Rus is that usually the very first spawning of the season in October or November is usually delayed compared to the other spawning in the season. This is something we observe every year, and we don't have an explanation for that yet. So except this first observation, which is a bit more variable, we have this correlation between light, accumulated light, and spawning start time. For the depth colonies, we only had one observation of a spawning colony at a greater depth, at 20-meter depth. So we only had one light and spawning colony data. What we observed is that these colonies spawned around after accumulated as much light as the colonies located at the surface in the shallow waters at that date. So we wanted to calculate the theoretical amount of light if this colony was located at the surface. So if this colony was located at the surface, it would have accumulated only much less light, and maybe it wouldn't have spawned. It would have accumulated less light than the other colonies in the shallow water that spawned. So here again, we only have 1 year of data for this light observation. So the idea is to put more loggers and add some light data to confirm these relationships, and also add data at greater depth to better understand what happens in the gradient of depth between 0 and 30 or more meters.
Charlotte, you have 5 minutes left.
Yeah, that's good. We also wanted to check the influence of temperature, because in other studies of other species, the studies demonstrated that temperature is very important to induce gamete maturation and spawning. So we collected the sea surface temperature from all dates and all sites that we had in our database. And we compared this to a logger that we had near the colonies. And what we collected from the models on the internet matches what we saw on the logger. So we decided to use all the data that we can find on the internet in specific websites that provide SST. And we collected all these data, temperature data, and matched this to our site and date for the spawning data that we had. And we detected a very strong correlation between sea surface temperature and spawning start time after sunrise for Porites Rus. As you have here, you have a significant linear correlation that shows that the Porites will tend to spawn earlier when the temperature is warmer. It works for at the site level from La Réunion in the Indian Ocean to the Pacific Islands, and it works also at the colony level. Here you have on the second graph the colonies recorded at the Verao site in Tahiti, and we have the same relationship. And what we can observe is that even at the colony level, when you consider one single colony, the trend is the same. Each colony can react to the changing temperature and spawn earlier when the water is warmer. So the take-home messages with these results here on Porites Rus is that we have a very, very strong and wide synchrony over more than 1,500 kilometers. We detected a delay that we have quantified between male and female spawning. We have a delay at higher depths, where the Porites Rus will spawn much later than at lower depths, where there is more light. And we have a delay at low temperatures. So when you have the temperature that warms up during the warm season, the coral will tend to spawn earlier, months after month. And we also observe when monitoring the tagged colonies that one colony is able to spawn 5 consecutive months and even 2 consecutive days, which is a lot for a single colony. So there are many further research avenues with Porites Rus and coral spawning in general. First, as I mentioned before, we need to add more data, especially for the depth and light relationship with a spawning time. We need to maybe investigate different wavelengths of the light spectrum, check the UVs, etc. We also need to check if temperature increase before the warm season can be a trigger of the start of the spawning season, as it is for the other coral species. We can also study whether there is a chemical transmission between male and female that induces female spawning that can detect that male are starting to spawn, or if everything is just about genetics and biological clocks determined by the genes. We would like to study a bit more the consecutive spawning variability, because sometimes we do observe consecutive spawning 2 days in a row, and sometimes we do not observe this. So many more things to look for. So now to get back to this workshop session, is Porites Rus a good candidate for restoration? That was my question, because in this presentation that we've had so far, we talk about a lot about acroporas and other coral species, but much less about Porites. Jamie mentioned some of them yesterday, that it is a bit more difficult because Porites are usually massive corals, and it's a bit more difficult to get fragments and garden them. What we think is that it's a very resistant coral overall. It can resist to many stresses, so it can be a good candidate for restoration because of this. We observed that Porites Rus colonies, like massive colonies that were relocated, it was like 2 years ago, because of construction activities on the shore, they spawned the very next month. So they were not affected at all by the relocation. We observed that branches that fall off can cement very fast to the substrate and continue to grow. And we also observed that it's very easy to fertilize the eggs and sperm. The issue that we have at the moment is that it is quite easy to collect, and you also have a lot of copepods going around the colonies during the spawning, and they eat the eggs. So it's not very easy to collect, but when it is possible, the eggs and sperm can be fertilized very easily. I'm not saying that the larvae can be easy to get, but at least fertilization is easy to make. So the next step would be maybe to test Porites Rus gardening techniques according to the different morphologies that it has. Rear larvae, try to get the larvae and keep the larvae, which can be beneficial for the genetic diversity. And of course, it is very important for us and for you and everybody in the world to continue the monitoring on this specific coral, because it's key to understanding how to better conserve and restore this species. Thanks a lot for listening. This is my contact information here if you have any question about this presentation, or if you want to know more about our research activities. Thank you.